Today, this is part of the regular Monday, I got to put a pitch in here, the Monday imaging lunch. So if you're interested in image processing, most Mondays there's a seminar of some sort going on and a mailing list to keep track of those. And so please feel free to come back for those. This is a special version of the image processing lunch because we have the pleasure of a guest coming from King's College London, Steve Niederer. And I know a number of you have been sort of tracking his visit and have meetings set up with him. Steve has a really interesting pedigree, and he started in a group that a number of us know and have had ties with in Auckland University. Peter Hunter is the sort of godfather of that group and leader of that group. Steve got his undergrad training with the lab in biomedical engineering, then moved to the UK and got his PhD at Oxford University, got very involved in imaging and software development, just an amazing breadth of projects. I think that's the thing that I notice most talking with him. There's not much he hasn't done or touched or has some ideas about, and he'll give us hopefully a bit of a sampling of that today. Since his time, since his PhD again at Oxford, he's moved to London, where he has a group at King's College in London. They're a very translational group, very tightly integrated into the clinical domain, and he's built a group of about a dozen people engaged in a wide range of projects, mostly to do with the heart, but mechanically, electrically, using imaging, using simulation, working with clinicians directly. It's a really impressive array. And so I'll stop talking and pass the baton to Steve, and I'm sure you'll be as fascinated as I've been the last couple of days with the amazing research he's accomplished. So, Steve.
Thanks a lot for having me, Rob. And it's been great being out here and experiencing powder snow for the first time as we went to ski in a storm yesterday. So this talks about the work that we're doing on developing kind of cardiac modeling, which is for a long time, certainly when I started my PhD, was a tool that was used for answering research questions and often preclinical work, and how we can kind of translate these type of models into being applications for doing research into drugs and to disease in clinical and application focus. So the way that we look at the heart is we start at electrical activation. So we have, oh dear. To move of a heart act. I'll just try that, yeah. Always an opportunity. There we go. So we have activation sweeping over the heart. Activation starts in the right atria, moving over the left atria, moving down into the ventricles, activating those. The models we represent start with this, or have this kind of 4-chamber organ scale detail. We then link down into models of the electrophysiology at the single cell level, representing the proteins which pass through the membrane, the sodium, the potassium, and calcium channels. Those feed into the calcium dynamics inside the cell for cytoplasm reticulum, leading to release of calcium, given a calcium signal, which leads into the sarcomere, leading to cross bridges binding, generating tension, and eventually giving rise to mechanical work in the heart. So this was our initial kind of framework that we looked at, and as we've done more and more work with different clinical groups, we had to expand this kind of relatively linear electrical to mechanics pathway to include more features. So one of the ones that we've more recently been interested in into the mitochondria, providing energy for these systems. We've had to look at reactive oxygen species, looking at including models of venting chemistry for mitochondrial DNA damage. We've done work on Takotsubo syndrome, as well as looking at some other kind of heart failure phenotypes. We've had to add in regulatory pathways, looking at beta-adreneric regulation. And because we've done work with computer scientists, someone always wants to make a model of CFD, and so we've done some CFD simulations as well, looking particularly at mitral valve dynamics. So I'm just going to give you a kind of a talk through a relatively succinct summary of kind of how we've progressed from these ideas, from the single cell through to the whole organ and then into the clinic. So this is the representation for the cardiomyocyte that we think about. So this is our cell membrane, our different proteins which are moving ions across that membrane and regulating calcium inside it. And mathematical models of the cell are fantastic because they provide a kind of a standard biophysically informed framework which we combine lots of data from multiple different groups. So if someone's characterized a protein in one lab and another protein in another lab, we can start to combine those and look at emergent behavior that's happening at the cellular scale. So it's a great way for combining data. Now the challenge is that the cell models don't like to give up the data that's been used to be put into them. So the way that most people go about making a model is they say, I want to make a model of a cell, They go into the literature, they find models of all the different proteins that they've wanted to, or find someone else's model which is similar. They take the parts they need and they put them all together to make their cell model. So what this means is the models don't like to give up data. So we start here with one of the first measurements and models of kind of this particular calcium binding protein, calsequestion, which was measurements done in rabbits. And then shortly after, well, 10 years after that, that data was used in a frog model of calcium dynamics. That was then used in 2 guinea pig models of calcium dynamics, the same parameter, same data. That was then used in a rat model and a mouse model and human models and so on and so forth. So one of the challenges we've seen with these complicated models and these large models is that they're very good for combining data, but they don't necessarily combine the relevant data or the best data every step of the way, and it can often be very hard to work out where that data has come from. So one of the visualization problems we've looked at is how we can visualize or look at what data goes into one of these models. So this here is a representation of a mathematical model of a mouse cardiomyocyte. The top trapezium, so the trapeziums represent mathematical models. Each of the different boxes represent a protein that were included in the model. And then the data that was used is represented by ellipses. But for some cases, such as, let's say, calmodulin, that's dependent on another model, which is dependent on another model, which then comes back to our data, which this one is from bovine. So we can start propagating through and looking at the broader range of species which are going into our mathematical model, and we can start to say, well, which parts of this model we'd like to improve if we would like to actually look at a mouse, as opposed to some kind of menagerie of species. So we were particularly interested in calcium dynamics, and just as the incredibly short primer on calcium dynamics, we have our cardiomyocyte, we have our calcium ions distributed around, calcium in the sarcoplasmic reticulum. We have our 4 major calcium handling proteins, L-type calcium channel, ryanine receptors, SERCA, and NCX, or sodium calcium exchanger. When the cell depolarizes, the L-type calcium channels will open, calcium will come into the cell, calcium will bind to the ryanine receptor, giving rise to calcium-induced calcium release, and depending who you ask, reverse mode sodium calcium exchange, bring a little bit of calcium in from the extracellular space. This will all bind to the sarcomeres, causing the generation of contraction. As this membrane depolarizes and the cell relaxes, calcium is extruded from the cell through sodium calcium exchanger, and also brought up into the sarcoplasmic reticulum through SERCA. And so we wanted to create more of a standardized workflow that we could make, and we could work with collaborators, or experimental collaborators, so people like worked with Pavel Svajcek, we worked with and Bill Lauch at the University of Oslo, for being able to make standardized experiments we could then use to characterize these models. So we have a caffeine transient-induced calcium flux, where all of the calcium is released out of the sarcoplasmic reticulum. That then is extruded through the cell through the sodium calcium exchanger. So because we know that the sodium calcium exchanger is going to be the dominant protein operating under this condition, we can then calculate the rate of change of calcium flux, capture the effects of buffering, and capture the calcium dependence of the ion flux through the sodium calcium exchanger. Once we have a characterization of the sodium calcium dependence of the sodium calcium exchanger, we can then do a field simulation of the calcium transient. We can subtract away, using the model of our sodium calcium exchanger, the effects of sodium calcium exchange, leaving just the SERCA flux, which we can then fit our model to SERCA. We can do voltage clamp measurements for the L-type calcium channel to make a mathematical model of the L-type calcium channel protein, and then we can combine we've got 3 of the 4 characterized, so we can subtract all of these fluxes from the field-stimulated calcium transient, allowing us to fit the ryanine receptor model. So we've used this approach for characterizing knockout models of SERCA, of looking at aortic banded models of heart failure in the rat heart. This is a relatively general way of creating of standardizing how you go about building a mathematical model of calcium dynamics. We can also go back to the literature to find other data which is more relevant or updated for characterizing electrophysiology, allowing us to simulate the action potential of the mouse myocyte in this case. And so we can look at how we've improved. And so we've focused on this particular section here, which was the calcium handling proteins, and we've been able to replace them all with experimental data. So we haven't solved all of the problems because lots of these measurements have been very difficult to do, and it's very hard to get people to be motivated to redo experiments just to make a model, characterize a model parameter. But at least we can tell where we were, we can tell where we've improved to, and we can identify what we might need to do next in the process. So we have these models of cells and of kind of protein scale physiology, and one of the areas we've looked at applying these is investigating a drug called doxorubicin. So doxorubicin was discovered in 1967 and it was used in early trials for cancer in 1969. In 1974, the meta-analysis of multiple cancer trials were put together, and it kind of was deemed that it was effective. And by 1976, it was found out that it was cardiotoxic. So it's still used today. It's a very, very common chemotherapy, but it has a number of challenges. So there are extreme delays in the toxicity effects. So there's a case study here of doxorubicin-induced cardiotoxicity 17 years after the drug was given. So the drug has well and truly left the system. So how has a drug which has left the system caused a change which has taken 17 years to manifest? We see a far greater survival rate now in cancer treatments, which is a great success, but that means that more patients are suffering from this toxicity than ever before, and that every few years people publish a new model for what the animal model should be for this particular toxicity, and so this remains continually controversial, so there is no real standard animal model for these systems. So when we're looking at making a mathematical model for this, we wanted to look at what are the, to evaluate some of the hypotheses that people have put forward based on observations to say which of these are kind of consistent with this long-standing toxicity. So patients take the drug, it goes to their heart, goes into their myocyte, and the main idea is that it predominantly affects the mitochondria. So we have our mitochondria here, and the very simple mitochondria is that our metabolite, the derivatives coming in from the cytosol into the TCA cycle. This is going to produce NADH, which is going to power this protein gradient through the electron transport chain. And when proteins fall down that proton gradient, they're going to lead to the generation of ATPase. So this is kind of the system that we're interested in. We're looking at 3 different mechanisms which people have proposed in kind of either discussions or in reviews that could possibly lead to this kind of cardiotoxicity from doxorubicin effects in the mitochondria. So the first one was that doxorubicin is known to bind to complex 1 and increase reactive oxygen species production. So potentially this is our source of cardiotoxicity. It's also known to bind to cardiolipin and alter the whole regulation of the electron transport chain. So that's another potential mechanism. But reactive oxygen species production can also disrupt the electron transport chain, and disruption of the electron transport chain can also cause Rus production. And doxorubicin is also known to alter the mitochondrial DNA, which is responsible for only a subset of expressions of proteins in the mitochondria. This can also cause electron transport chain, but Rus production can also alter the mitochondrial DNA. So we have a number of feedback loops which mean all these mechanisms are potentially interlinked. And the question we had, or as engineers looking at this, we thought if something is going to have a long, slow process of being observed, it's likely to be to do some kind of feedback mechanism. And so we had 2 potential feedback mechanism loops within this system through the electron transport chain and Rus production, and also through mitochondrial DNA damage. So we used the Hopkins mitochondrial energetics model, which had all the systems we wanted in, including in particular the Rus production and scavenging system. And then we looked at how we could represent these different mechanisms within that model. So we had complex 1, we could just shift the flux of protons through complex 1 to have more Rus production. For cardiolipin, there were dose response curves for each of the complexes, which we could then use to calculate how doxorubicin might alter the flux through those proteins. And then there was no model of mitochondrial DNA regulation. So we wanted to look at how we could make a relatively crude model of mitochondrial DNA regulation, where we added in a mitochondrial DNA repair, which we thought would be enzymatic and dependent on the amount of damage done to the mitochondrial DNA, that would have a basal level of reactive oxygen species damage to the mitochondrial DNA rate, and would also add in a component for capturing how doxorubicin would directly affect the mitochondrial DNA. And so our belief was that if any of these mechanisms or combinations was possible, it would have to cause either a shift in the system or a steady decline in the system. So if it causes a shift in the system, then we've gone from one state to another, and would assume that the second state that will be moved into might be more susceptible to cardiac disease. So when we looked at including mechanism 1 and 2, so the complex 1 effects and cardiolipin, we saw no fixed shift in the system. So it was hard to reconcile either of these hypotheses that people proposed before as being an explanation for the long-term change in the mitochondrial function. Once we add in the mitochondrial DNA damage, though, however, when we fit to kind of measurements done over 44 weeks in a rat model, we could then see that we had a systemic change in the mitochondrial function. So after we'd given our dose of doxorubicin, the model was then unable to recover. So it was then in a steady state of decline if enough doxorubicin was given, consistent with the clinical observations. So the reason for this largely comes down to the fact that our mitochondrial DNA repair equation had a saturation effect. So we have that if you have a high level of mitochondrial DNA, more than you would have normally, your mitochondrial DNA repair is very downregulated or not very active, and the basal reactive oxygen species will actually be sufficient to cause your mitochondrial DNA to decrease. Eventually, the repair and the background reactive oxygen species damage will balance, and you'll reach an equilibrium point, as we can kind of see in our phase plot here. And at some point below that, you've got enough repair such that you can recover. So you'll move within this region, you can recover back to your stable point. But at some point, you pass the point of no return, where so much reactive oxygen species have been produced that your recovery mechanism can no longer bring your mitochondria back to a physiological state. And if that happens, you reach this unstable or terminal decline region. So this was where we could take a series of hypotheses, which have been all done in independent, relatively narrow measurements, interpret them within the context of the whole mitochondrial system, and come up with a plausible explanation of how doxorubicin could result in a 17 or 1 year delay in the toxicity phenotype that has been observed. So the other system that we look at is modeling at the cell level, is modeling tension generation. So we have before we have the cell membrane depolarizes, calcium comes in, calcium is released, or through the alpha calcium channels. Calcium is released from the sarcoplasmic reticulum. It comes down, it binds to troponin C. Troponin C causes tropomyosin to move, revealing the actin binding sites, and our cross bridges can then bind and generate tension. So this is a relatively straightforward linear progression. Unfortunately, or fortunately, depending on your perspective, there's a lot of feedback mechanisms. So the cross bridge binding will be velocity and length dependent. Tropomyosin kinetics have been deemed to be length dependent, and that we have a tension or cross bridge dependent binding of calcium to troponin C. And all of these dependencies are going to be dependent, again, on how many cross bridges are bound and how much the muscle is deforming. So it's a very non-linear feedback system. And a lot of work has been done on this, and a lot of it has been done in rat or in rabbit models. But if we wanted to move into the clinic, we really wanted to look at how we could make mathematical models of tension generation based on detailed measurements made from human tissue. And the problem with healthy human tissue is that most people are using it. So we had a collaboration set up with Professor Crispar dos Remedios in Sydney, and they were had an Australian biobank, and they would send us cardiac tissue to London, where John Kentish's lab and Dr. Seo-Jin Ho would do single cell measurements of isolated human myocytes. We'd propose structures for representing the different protein kinetics, so protein maps or protein-protein interaction schematics, and then we'd derive equations for those, and then we'd fit those to the data. And so the data we have, and this is something that was a harsh reality for us. So I've done a lot of work using literature data, which is always based on representative traces. And when you start to collect your own data, you realize that noise is really large and that it can be quite challenging to fit. So we're able to change different lengths. We can look at steady state force curves. We can do rapid length steps where we deconnect all of the cross bridges, and we can watch as they recover at a steady calcium and length. We can do short length perturbations up and down, look at kind of perturbing the cross bridges but not having them all detach, and looking at how that changes their cross bridge kinetics as they enter an equilibrium state. So we have our schematic for how we believe or how we're going to represent the cross bridge kinetics. So we start off with in an unbound state where we are actin polymer and a tropomyosin molecule blocking those binding sites, and troponin I kind of remote from interacting with tropomyosin. When calcium binds to troponin C, troponin I moves out of the way, binding troponin C into a pocket which becomes revealed when calcium binds. Tropomyosin moves out of the way. Our cross bridge is still unbound, but once it has actin sites available, it can then move into its first weakly bound state, and then a power stroke strongly bound state where it's generating tension before it can detach and then retach again, cycling round. So the measurements that we performed were length step measurements. We were able to calculate both the elastic and viscous passive properties of the cell. We could do force length measurements, where we were then able to look at making measurements of how the binding or the calcium sensitivity of the muscle changed as you altered length. So these measurements are steady states. We have calcium, which is the signal for generating tension along the x-axis, and on the y-axis we have the amount of tension that's been generated in kilopascals, and we can look at that at 1.8, 2, and 2.2 micrometer sarcomere lengths. And we also have a fair level of variability in our models. So each one of these is a different cell, and so we see a trend where these are kind of moving up, but we still see a fair level of variability within those calcium FPCA curves between the cells. We can also look at doing stretch perturbations, where we're looking at, as you stretch the cell, the cross bridges don't necessarily detach if you do a very small stretch, but they get moved into a distorted state. And as they recover from that distorted state, they unbind and then rebind, and that gives us this complex kind of biphasic tension transient that we see from the average curves, and that gives us an idea of being able to characterize how distortion might be in velocity, might be affecting the cross-bridge kinetics, and how those kinetics are going to be, and gives us information for characterizing cross-bridge cycling parameters. And so one of the challenges that we have with experiments is that, particularly for tension generation, is often these measurements are done on isolated myocytes. This is where the membrane of the cell has been permeabilized so that you're able to control the intracellular calcium concentration by changing your bath solution. The difficulty is that this demonstrably changes some of the properties of the sarcomere. So this is a kind of a measurement of a calcium transient from a human trabeculi. We see it goes from about 0.15 micromoles up to 0.6 micromoles. And we can look at the range of calcium that covers on our steady state forced calcium curves, and that's this region here. So if we feed this calcium transient into our forced calcium curves, what we predict is that we generate zero tension, which is in many ways incompatible with life. So we then had to go back and we did a sensitivity analysis, we looked at the literature to find out which of these kind of parameters are the most likely to be altered by the skinning process. We then characterized what was the type of tension transient we would expect to see in human muscle, and then we could refit the model to characterize which parameters we had altered to be able to recapture the trabecular scale tension transients that we'd expect to see in healthy mouth tissue. Now, this is not ideal, but unfortunately, having spoken to experimental people, and I'm happy if anyone can suggest a solution, characterizing the effects of skinning on tension generation is very, very difficult. So the other thing that we then do is, having gone from our single cell model to a trabeculi, we can then put these models into whole organ heart models. So these are healthy controls data sets, and so we get our healthy control anatomies from the emergency room. So people come in with acute chest pain, and in our hospital they have a CT image, and then if there's nothing wrong with them because they had indigestion and weren't having a heart attack, then we can use their CT images for characterizing our healthy control patient group. And we can then fit our model in, and we can look at putting in the attack cell model. So when we put in the model that we have for the attacked myocyte, we're able to get a nice healthy looking tension transient. We get a good ejection fraction down at about 50%. So this meant that we had a model that we felt we were happy with, that matched the cellular data, but was also compatible with the organ scale phenotypes we would expect to see for healthy patients. This kind of provides us the framework that we have going from the single cell electrophysiology to the energetics, to the mechanics, and now we look at how we can start bringing this type of cellular modeling together into more of a clinical and organ scale simulations. So one of the things we do a lot of work on has been desynchronous heart failure. I know that some work's been done here in this area. This is where we have a healthy heart, we have electrical activation in the atria, starting on the right, going to the left, for rapidly activating the right and left ventricles through the Purkinje network. And then desynchronous heart failure will have the left-hand side, the Purkinje network will be blocked, it will be compromised in some way, and you will have a slow electrical activation from the right to left moving through the myocardium. And this is going to cause desynchronous, inefficient, and ineffective contraction. The way this is treated is we put in two pacing leads, one through the coronary veins to pace the left ventricle, and one into the right ventricle to pace, or right through the right ventricle cavity or apex to pace the right ventricle of the heart. And this augments the intrinsic activation system such that we get return to ideally a rapid and efficient contraction. So what we have here is a kind of an idea. We would like to make patient-specific models for these patients to identify what patients might respond best or how we could best treat them. So we do this using MRI for these cases where we had a high-resolution anatomical MRI. We could then segment out the myocardium for the tissue. We then use a mathematical description for the anatomy for a template heart, and then we can register that template mathematical topology to that individual patient's heart, giving us a mathematical description of their anatomy that we can then solve our partial differential equations for electrophysiology and mechanics over. At the moment, it's not possible to readily get fiber information. So we have the heart has a fiber structure which kind of varies both apically to basally, from the bottom of the heart to the top. It varies transmurally. It changes in the septum and in the right ventricle. And so we use a rule-based a rule-based method for generating microstructure that is personalized to the anatomy that has come from histological data. Once we have this model of with the fibers and with the anatomy, we can then start to look at solving electrophysiology simulations. So here we're activating from the right ventricle. This particular model had about 30 million cell models. This is where we're able to have all the sodium channels, potassium channels, calcium, intracellular calcium regulation is all represented in this model, and it can vary spatially in different regions across the heart. We'd also like to look at, or we do include the mechanics of the heart. So we have cine MRI combined with tagged MRI, and then we can track the motion from each of the images, and then we can embed one of our models describing the anatomy of the heart within that image warping field, allowing us to track the material points and displacements of those material points throughout the cardiac cycle. And so this is the type of information which allows us to calculate strain in the heart, which we can then use to drive and fit our models of mechanics. So with these models of electrics, mechanics, we can then make measurements of the pressure transient in the left ventricle, both at baseline and during pacing protocols. And so the yellow line is the experimental measurement, the blue line comes from the model. Here the yellow boxes are coming from MRI and the blue line is coming from the model. And we can also make predictions from things that we didn't measure. So here we have the yellow spheres representing an infarct region of the heart which aren't contracting. The blue spheres represent regions of the heart which are doing positive work, so they're doing work on the surrounding myocardium. So during ejection, you want as much of this as possible. They're going to be what's pushing the blood up and out of the heart. And then in relaxation, we have the red spheres where the myocardium is then doing work on that particular piece of tissue. And it's the balance. Ideally, you'd have everything be blue and everything be red, so we have a synchronous contraction and synchronous relaxation, and that's what's really been treated with the CRT patients. So one of the questions that we have is what's important. So we have lots of information for these patients. We have our MRI, or CT, pressure measurements, echo, we have X-ray, we have doctor's notes, we have pressure catheters, and we now have a model which we can represent a lot of features, and we can do some studies to try and identify what particular attributes of this patient is important for its response to CRT. So here is kind of a list of all the model's parameters. So we have amount of peak tension, tension generation, how fast tension relaxes, how fast it develops, end-diastolic pressure, stiffness, conductivity parameters. And we can go through perturbing each of these parameters and looking how well the patient or in the model responds to turning CRT on or turning CRT off. And we find that we get the most important parameter for determining if a patient will respond to CRT was this length dependence of active tension. And so this is analogous to the Frank-Starling mechanism in the heart. So in CRT, when the heart, for the ACE, so during desynchronous heart failure, when the septum is first activated, it contracts early. And because the heart is in a period of isovolumetric contraction, so that the volume can't change, if the septum is contracting, the lateral wall is going to be extended. So we see this shortening of the septum and prolongation of the lateral wall, or the remote wall of the heart. So kind of in a more schematic sense, we have our septum, which is going to be activated early, our lateral wall, which is going to be activated late, and as our length dependence of tension increases, we're going to see a faster rate of tension development of a longer muscle, and a shorter rate of tension development, a slower rate of tension development for shorter muscle. So faster, slower. So that when the septum activates, we can see that tension is going to develop in the septum while the lateral wall will remain quiescent. That will cause the septum to shorten while the lateral wall will extend. When the activation eventually reaches the lateral wall, however, the lateral wall is now at a longer length, so its tension generation is going to be faster, while the septum is a shorter length, so its tension generation is going to be slower. So despite having an asynchronous electrical activation, if you have a healthy Frank-Starling mechanism, you're going to see a synchronous contraction in the heart. However, if you have an unhealthy Frank-Starling mechanism and you're not going to see this change in tension development with length, your mechanical or tension development will remain as asynchronous as your electrical activation. And this substrate here, where we have these two lines running in parallel, is kind of the ideal substrate for CRT, where you can alter the activation time and bring these lines to be in the same region. So the conclusion from this study was trying to identify what were the possible mechanisms would explain why patients might respond to CRT or not. And one of them was that a compromised Frank-Starling law would increase the coupling between the electrical and mechanical desynchrony and make these patients more susceptible to a response to CRT. So the other area of the heart, which we haven't done as much work on, but it's kind of a new area for us, is looking at the atria. And so we're really the atrial electrophysiologists that we have in the hospital are kind of making measurements of anatomy with the MRI. They make structural remodeling measurements with late enhancement imaging, functional modeling with electroanatomical mapping devices. We can measure the mechanics either from the cine images, and also making hemodynamic measurements with pressure. And what we found when we spoke to them was that all of these measurements are made, but they're never really interpreted all at the same time in an integrated sense, certainly not in our center. And I know that's something that you guys certainly work on here. So the way this started was that I got a data set. I segmented out the data set. It was a CT image. I made a 3D model of the heart with tissue. I put a cell model in. I made a simulation. I said, look, we can now, looking at the membrane potential here, we can simulate an arrhythmia. This is exciting. It has color. The color is moving. So normally when I sell that to a cardiologist, that's definitely a win. And the cardiologist said, and this is what I learned from kind of, I share an office with the director of EP cardiology for the St. Thomas's Hospital. And I showed this to him, and I thought he'd be reasonably happy with this. And he said, so you've characterized the anatomy. Can you tell me how thick the wall is? I was like, that seems dull, and I don't really care. But he convinced me with the video. And so when you're doing atrial fibrillation, you often have to do either cryoablate, which is with ice, or kind of a radiofrequency ablate, where you deliver energy to try and create lesions in the wall of the atria. And you want your lesion to be transmural. And so what we're looking at here is someone receiving a cryoablation, and we're looking down their esophagus, and the atria is somewhere here, and we're seeing the formation of an ice ball inside the esophagus during the ablation procedure. So this is not what you'd like to see. So you don't want to be delivering so much energy that you're damaging the surrounding tissue, but you want to deliver enough energy that you are creating a transmural lesion. And it would be nice to know how thick the wall was, because that could be useful information for titrating the energy. So we went away with the naive idea that we knew what thickness meant, and then we had to realize that we didn't. So we thought thickness, that's a normal, we have one surface, we'll do a normal prediction. When we hit the other surface, that'll be our thickness measurement. And that works fine for position Y, but for position X, you have a huge artifact because you go off into oblivion between two surfaces. And that's kind of an example where you might see that from a CT image. So we thought we'd look at the shortest distance between two surfaces. And again, for position X, that works well. But position Y, this surface is actually closer than this surface, so you have an artifact where you're kind of going over a blood pool or some space, which you shouldn't. So one thing that we've learned in cardiac image processing is that the brain people tend to be smarter and better than we are. And we believe that's largely because the brain doesn't move, and so it's a trivial problem. So that had brain cortical thickness for some time, and looking at how you can calculate brain cortical thickness. And one of the ways you can do this is you can solve a Laplace problem between the two surfaces. This creates a series of smooth isosurfaces between each of the two surfaces you have, and you can calculate effectively field lines moving orthogonally to these isosurfaces, and calculating the distance of those field lines gives you a really nice way of calculating what thickness is. And it gives you a unique one-to-one mapping between each of the surfaces. And so mathematically, this was quite a nice solution. So we then went about implementing this. We had our CT image plan. We looked at using distributions of the ventricular myocardium Hounsfield unit to determine what the Hounsfield unit distributions should be in the atria. We could then segment out the atria anatomy. We could make a high-resolution mesh that we have a lot of experience with for our electrophysiology simulations. And then we were able to create what were some of the first, or possibly the first, I think, maps of thicknesses across the whole atria. And the previous work on atrial thickness has tended to be with calipers within a 2D slice. So you have a very limited number of points that you're making measurements on, and you're also making all those measurements within the imaging planes, which are dictated to you by the scanner coordinates, not by some kind of orthogonal distance. So we're very happy with that. However, we were told that we had to really validate this, so we couldn't just believe that it would work. So that was a reasonable, so we started off with 3D printing. So we made our kind of pseudo atria, which doesn't have an appendage, which is one of its many shortcomings, but it does have different thicknesses in all of the pulmonary veins and 8 different thicknesses around each in each different region. We could then do a CT of that, of our phantom. We could then perform our thickness measurements on the phantom, and then perform our thickness measurements on the images that were derived of that phantom and show a good match. Unfortunately, again, doing measurements or validation on patient hearts is very difficult. So we had 10 living patients who had cardiac CTs who were having an ablation of the left atrial appendage ostium. And So we could make measurements of their osteums, and then we could compare those measurements we made in the osteum with cadaveric measurements of 10 age and disease-matched patients, and show that we had a good match on average with the thicknesses in the osteum in the diseased patients, with the cadaveric patients, and in the living patients. And then more recently, we've done some porcine studies where we've been able to show a good correlation between the thickness measurements we've made in histology with the measurements we've made in CT. One of the challenges we've had in this particular case is that the atria isn't unloaded when it's inside the heart. So it's actually, it is a pressure vessel, so it is deformed. It is there'll be some motion. You don't know if you're in a perfectly relaxed state. And when you take it out, you've now cut it out, you've now changed the structure. So we expect to see a correlation between the thickness in different regions, but not necessarily a one-to-one match for those regions. So now we found a data set out of Budapest where they've done CTs on 180 healthy individuals. And so we've now done our wall thickness calculations on those 180 individuals. We've been able to register those all to an atlas, and we can now generate kind of mean atlas maps of wall thicknesses across the atria in different regions. And the reason we wanted to do this was so that we could first have an idea about which regions are thin, which regions are thick in a healthy person, and so that will give us an idea when we look at pathological people how deviant or far away they are from normal. So we'd gone through this process of starting with a fancy model which had lots of colors, and we'd come to the end of this process and we'd become a lot better at making models of the anatomy of the atria. So the other things that we had to start thinking about was how could we add in, if we want to make kind of more detailed models to capture the mechanics and the electrics of the atria, was thinking about the microtissue structure. So the measured DT-MRI is hard to do in a healthy heart or in a control heart. So I'm sure you guys are doing DT-MRI scans on healthy controls or patients, but the image quality is not great in the ventricles. In the atria, it's likely to be challenging for some time into the near future. So we decided that we weren't going to try and image fibers in living patients, and so we then had to find a rule-based method that we could take histological data and project that into our atrial anatomies. So we did this by defining 151 different regions across the atria, and we were working with Professor Yin Ho, who's basically written all the books on atrial anatomy and is at Imperial College London. So we could go across the river and have a chat to her, and she dictated to us what she thought from her experience with histology were the mean fiber fields that we needed to include. So we made 151 different regions, and manually putting those onto each atria was going to be a pain. So we labeled all of those on an atlas, and then we can register our new patient atria with an atlas. We can then do a projection of those landmark points from our atlas onto the patient's atria, and then we can generate all the regions using shortest path calculations, and then we use a series of Laplace solves to generate smooth fiber fields across the different regions of the atria, both on the endocardium and the epicardial surfaces. So when we do this, we can then start to look at simulating electrophysiology, not in the models that we showed you before, which were generally generated without any fiber data, but we can now make these models, which are very detailed. These have, for people who solve monodomain equations, these have a 125 micron mesh resolution. So I think we have on the order of 100 million elements per mesh. So we get very converged solutions. We get nice activation waves. We have that level of resolution so that in regions of wall thickness where it's only 0.5 millimeters thick, we have at least 4 or 5 elements across the wall to capture any transmural variation. And this is the most recent work. And so this is not the best picture, but these we're looking at, we've done passive inflation of each of these myocardiums. So we're now looking at solving the nonlinear mechanics using a transversely isotropic constitutive law. We provide a passive inflation, and we're looking at here at the first principal stresses in the myocardiums, both from for different views, kind of going up and down. And so we start to see that we see these distribution of stresses, which are both determined by the curvature of the atria, but also by the heterogeneity of thickness in the atria. So there's a question that we have: if stress is going to be driving remodeling and possibly high regions of strain, how does that relate? How does the curvature of the atria that we see in these patients and the thickness distributions that we see dictate what stress distributions that we're going to be seeing in these patients? So this is kind of where we've got to with these high resolution models and kind of deriving information from images. The other area that we're very interested in is how we can derive mathematical models from functional data. So instead of just taking an image and saying we're going to use representative physiology, we've started to look at how we can make models that capture the physiology and the heterogeneity of physiology in individual patients. So this is your kind of standard, not standard, this is your cath lab image, your X-ray fluoroscopy, X-ray image, and we've got an overlaid anatomy of the atria on top of that fluoroscopy. And here we have a decapolar catheter. As the name suggests, it's an electrical catheter which has 10 electrodes. So we make a 1D mathematical model of this decapolar catheter. And the great thing about these is they're used in pretty much every atrial fibrillation ablation study, and so we don't have to pay for them. So it's a pro tip. So we have a 1D model of the decapolar catheter, and this gets placed in the atria, in different regions in the atria, and we can stimulate the atria from the two central electrodes E5 and E6, and then we can watch the activation wave as it propagates either up towards E10 or down towards E1. And because we have enough electrodes, we can calculate what the conduction velocity is. We can do an S1, S2 pacing protocol where we pace at one pacing rate, so 300 milliseconds, 400 milliseconds, 500 milliseconds, and then we do an extra beat at a different rate for our S2. We vary that S2 rate to build up conduction velocity restitution curves, and as we bring that the second beat down, we can look for where the electrical stimulation becomes refractory, so it no longer propagates, and that gives us a way to estimate what the effective refractory period are for these patients. And so we created a cell model, which was an adaptation of the Mitchell-Schaefer cell model. It's a very simple electrophysiological model, but we effectively have two parameters, tau in and tau close, which characterize the opening and closing of the fast sodium channels, and tau out and tau open, which characterize the kinetics for the slower potassium channels, which govern repolarization. And we also have a conductivity. So we have 5 parameters for these models. And so at the moment when we're making these models, we basically went through simulating activation in this 1D model, and we did that with 500,000 different parameter permutations. So once you've done 500,000 parameter permutations, you've now gone from a optimization problem to a simple lookup table to find out what your best parameter fit is once you get data. So this means that we can go into the clinic, we can make our CV restitution curves for different S1 pacing protocols, we can measure our effective refractory period restitution, and with a matter of minutes, we can have identified the optimal cell model for this particular patient, for this particular region in their atria. So once you've made a mathematical model characterizing the conduction velocity and effective refractory period restitution for a particular region, you can combine those to start to think a little bit more about what type of activation patterns can these parts of the tissue, can they support? And so to look at this, I don't know what's going on. So to look at this, we can set up 2D sheet models where we've made measurements in 5 different regions of different atria, and we've been able to then simulate, are these regions of the atria able to support a sustained spiral wave based on their conduction velocity restitution properties and on their effective refractory period restitution properties. So some of you may be familiar with Sanjeev Narayan's work where he's trying to identify regions of the tissue which are supporting spirals. That requires a 2,000 pound or $3,000 basket catheter, whereas this is kind of coming up a way of saying, can we look at the electrophysiology of the local tissue properties, and from that information, identify where we might want to be ablating, or which regions can support a stable spiral wave, which regions are going to break up. So this was an interesting idea which we're now continuing to pursue. We're not quite confident enough to go straight to humans, so we're now start looking at small animal work for this. But this was making models of the electrophysiology at a single point, and it's interesting, but it doesn't necessarily tell you the whole picture, and it makes it hard to kind of link this back in with the anatomy of the patient. So we then went on and looked at the pentarate catheter. So this is a kind of a nice catheter that we like in our hospital. It has 20 electrodes, they're nice small electrodes, and we make measurements in the left atria with this catheter as we pace either from the high right atrium or from the coronary sinus, again doing this S1, S2 protocol. And then we can make activation maps across the whole atria at different S1, S2 pacing rates. And we can, so these give us kind of these distributions of conduction or of activation times. So this is where we're only using the measurements from the coronary sinus data. And so we can then use that to fit our models locally, as using the same approach as we did with the decapolar catheter. And then we can interpolate and extrapolate the parameters that we come from our models over the whole of the atrium. We can then predict what the high right atrium pacing pattern will look like for these patients. And so this wasn't data that was used to constrain the model. We make our predictions, and then we can evaluate that to give us an error distribution to give us an idea of how well our model is able to capture the electrophysiology at different pacing rates from a different pacing region. So we're able to both make the model and validate the model from a reasonably succinct data set. And once we do this, We can then start looking at simulating arrhythmias where we have the anatomy, which is coming out of the electrical anatomical mapping system, but also where we've captured the heterogeneity of the patient electrophysiology, not just assuming that it's going to be homogeneous or derived just from the images. And so we then start getting these nice images, and this is kind of where we're up to this, and we're now looking at kind of studies about effective size and multiple wavelets and where do spirals live. So this work has all been about, that I've presented so far, has been about how we've used our models to kind of understand the patient physiology, how we can use it to interpret clinical information, how we can get more information out of the clinic, but would really like to move these ideas from interpreting the data to kind of applications. And so this has been a process that we've been thinking about for a long time, and so one of the first things we had to do was convince from our collaborator standpoint that the code that we were writing was actually solving the equations that we'd written down. So we've done two things there. We've looked at verification problems for electrophysiology. So this was a problem which we defined an incredibly dull problem, but lots of people could solve. So we had 11 different groups solving this model with different spatial resolutions, different temporal resolutions, and the result has been that I think it's been cited over 100 times, where people have used this for computer science departments can now get involved and say, we've made our model, it works, it's as good as the best models that are available. And it's allowed the people who are making kind of who've been in the field a little bit longer to start to test new algorithms and say, we get the right answer, but we can get it with less power, less time, with sparser matrices. And so we did the electrophysiology, that was back in, I think, 2011, and more recently we've just done cardiac mechanics, where again we had 10 different groups. This was a combination of industry groups, so we had IBM involved in this one, as well as a number of other groups from Norway, looking at what type of elements we might have, how do you define a mechanics problem so it is reproducible. So a whole lot of relatively, I would say, not exciting, but kind of very important elements that you need to make your work reproducible, make it standardized, and if you're going to go through a regulatory pathway, this is the type of stuff that the FDA is certainly going to be interested in. So we also wanted to look at how we could make our models faster and simpler and know what information we needed to have. So for electrophysiology models, we had people that said that scar might be important for the activation pattern, that anterior or posterior functional block might be important. This is where the functional block occurs that you can't see on any MRI, that we might have slow conduction in the septum or fast conduction in the endocardium. And so we worked with a group in Maastricht with Fritz Prinzen and Kevin Veruni, and they sent us 14 cases where they'd done MRI and they'd also done epicardial activation map measurements, where they'd used a NAVEX system, they'd made measures of activation time in the epicardium of the left ventricle, and then we could make simulations based on pre-procedural data, adding in each of these different mechanisms and seeing which combination or individual mechanisms were required for us to best be able to model the activation patterns that were measured from the MRI and the epicardial activation. And once we'd done that, we then spoke to our cardiologists, and they said they would do the same. So we then had 4 validation cases where we used cardiac CT, so a different modality. We were able to pull out the coronary veins. We could then do the same, using the same electroanatomical mapping system, make measurements of the epicardial activation times, and validate that the models that we created using our rule-based formulas, using only non-invasive pre-procedural data, could give us a reasonable estimate of what the activation pattern would be in these patients. So the benefit of being able to make a model of the activation system is not just that you get a better idea of what the activation pattern might be in different regions of the heart, but it also means you can perturb the heart. So we have body surface potential mapping, which allows us to characterize the state of the patient, but with simulations we can start to predict how that patient might respond under different conditions, and that we can then use in our procedures to say, if we pace here, what will the activation pattern be, and use how the patient is expected to respond to optimize the therapy, not how the patient is. So moving forward in that direction, we've started looking at how we can use a combination of motion and simulation for characterizing where we should be pacing patients' hearts after CRT. So we have our CT image, we've got our MITK-based software, which was basically, we wanted a software that was designed so that the cardiologists could make the models and do the analysis. And the reason we wanted them to do that was because if we give them a result, they believe it to be true and completely ignore all of the caveats that we give them. Whereas if they make the model themselves, they're at least aware of that this segmentation wasn't right, this might be a shortcoming, this might be something that we need to consider. So we can do motion tracking from the CT, we can then pull out what strain is, we can look at area strain, circumferential strain, or longitudinal strain. This is just feature tracking on the endocardium. We can then plot on our AHA segment plot the different curves for the strains, and there's different regions. We can look for akinetic regions, which we might not want to pace because they might be indicative of scar. We can look for latest contracting regions that we might want to pace as a region, as a target. But we can also use our rule-based method to generate very rapidly, this takes about half an hour, to make a model of the patient's electrophysiology, where we can then simulate what the electrical activation pattern will be in these patients. We can then go through and simulate all the different possible activation regions they could have for the AHA segments, identify which one of those is going to cause the most synchronous activation, and then point that out to the cardiologist on the AHA map. So we have two ways of identifying where we might want to pace the heart based on the AHA map, and we can then overlay during the CRT pacing this is an example one, so where we've put in the AHA segment regions overlaid on top of the X-ray. So we can say if we want to pace in AHA segment 16, look for the green region, that's where you need to be putting the electrode. So this is where we're up to with our patient translation. At the moment, we've done 2 dry runs, and I think next week we have a patient signed up to do the first kind of live case, so that should be quite exciting. And the last thing I wanted to mention was kind of where I think the modeling, or at least our modeling, is going. And so for a long time, we've been looking at the atria and the ventricles as 2 separate entities. And as we've kind of studied them more, and as we've started looking at diseases such as atrial fibrillation with heart failure, or diastolic or HFpEF, or heart failure with preserved ejection fraction, or diastolic heart failure, the need to link both the atria and the ventricles together became more and more important. So we're using our CT images, we're generating 4-chamber models of the heart, so we have both the atria, both the ventricles. We can make high-resolution meshes of these atria. We can then generate rule-based fibers across all the different chambers, and then we can use those for our simulations, where we can both simulate the electrics and where we're improving our simulations of the mechanics. This is very much work in progress, where I think that the ability to link the activation pattern and the mechanics of the atria through the ventricle is an area, under-researched area at this stage. So this work has been done by a lot of people. So this is kind of the members of my group who are working on this, and the past members. We have a lot of industry support from device, imaging, and drug companies, and in particular, Mark O'Neill and Aldo Rinaldi are the 2 cardiologists who I work with very, very closely, and to be responsible for basically enabling all the clinical work that we do. Thank you. I'm open for questions.
Did you do any studies to convince the clinicians that by looking at the shape with your modeling will really make them, any comparator studies with what we have in clinic right now? With the echocardiography and the MRI, not just use them in order to get better and use these in order to get the modeling that you need, and couple these with your cellular studies. That's great.
Yep.
Did you really go to compare in any study, or is this something that you are thinking that you and your group, in collaboration with cardiovascular clinicians, can do? Because when we present these new concepts and new ideas in the clinic, that's what the busy clinician is going to ask you. These fancy images, he will tell us, or she will tell us, how are they going to make me manage my patients better compared to my conventional echocardiography, or my advanced echocardiography, and my advanced MRI?
So there's a number of parts that are possibly there to that question. So one is that have we validated our models against images? And so we have done that. So I gave a relatively whirlwind tour. So we have compared the overlaid with the predicted motion that we have from the models, and overlaid those on CineMRI to show that the models were.
The validation was great.
Okay, so happy with that.
Do you want to go to the? You know when?
So what's my pitch to say why give up on echo? Why try something fancier?
Great.
Yep. So the reason for that is that I think that for some of the diseases we have is normally. For the atrial fibrillation, for example, the success rate's not great. For CRT, the success rate's pretty good in some patients, it's not so great in others. And I think it's where you have patients where they're looking at everything they have and it's not delivering, that there's a gap there, you can say there's a gap that we can fill with fancy technologies which might be able to basically deliver some improvement.
The premise is there.
Yep.
I totally agree with the rationale. We need to do better, we have to the plan to convince, and my point is,
so what is my plan to convince someone?
There.
Yep.
You show clinical predictive markers.
So for the CRT work, the idea will be to do a clinical study and say that we can, so that in the first instance for clinical CRT, we're using acute hemodynamic response. So what is the percentage change in pressure generated, and can we predict which region will give that percentage change in pressure generated with different pacing? And so if we can predict that, we say you pace in this region, you'll get the best response. We've shown that, then I think that's something which would be more compelling to a cardiologist as a in the first instance, a harder and more clinical endpoint that we could work towards.
And you can impact the other disease processes as well here. You can impact the way that ICU physicians think, and surgeons, cardiac surgeons, not just the cardiologists. Our community needs a lot of help in these to understand ventricular mechanics. So that's great. And my other question was on the PV loops that you showed.
Yep.
Can you elaborate a little bit there on how did you manage to connect the two?
To connect the pressure and the volume. So which PV loop? So there's a pressure volume that I loop, which I showed, which are purely simulated. And the purely simulated ones are very easy to connect because the model gives you pressure and volume at the same time. For the other ones, for the way we normally do that is we look at, we have an echo that has an ECG gated on it, which will be off the R wave. And that if we can record our pressure transients at the same time onto it as the ECG on a dual device, we can then synchronize through the R wave. There are challenges because the echo is not performed on the same day, in the same position, at the same level of anesthetics. And so if we do have some variation on that, we tend to scale the time transient of the echo because we want to preserve that pressure measurement because that's what's been used predominantly for CRT for the acute hemodynamic response. I would not say that was a perfect, that's a compromise, I'm fully aware of that. But we've tried to do echoes in the cath lab, and the patient has a terrible position, it takes forever, and you get not great images. If you guys are able to do that, then I'm more than happy to get some advice on how we could get better echoes in the cath lab.
Yeah, Hi, Jim Feng. I'm a cardiologist like Dr. Dracos, and I enjoyed your talk. A couple of questions that plays on his last question. Pressure, how are you measuring the pressure input? Is it invasively, non-invasively? You have beautiful time volume relationships.
It's invasively.
Invasive.
So it's a
catheter in the
retrograde femoral access through the aorta, and down it's a radi wire from, I think that will be now Abbott.
Okay. Yeah, that's just, fundamentally when we do PV loops, which is arguably the best way we like to study the mechanics of the heart, getting the pressure input is the big challenge for real-time studies.
Yep. And
a related phenomenon is how have you imaged the vascular tree? Obviously, ventricular vascular coupling is a huge determinant of myocardial efficiency. Do you actually also dynamically assess the volume and compliance of the vascular tree?
No, so to be honest, we have not considered that. That is so we've looked at the mechanics, and we've assumed that the sarcomere force generation is kind of the principal driver, and that although other things are important, they will be secondary effects after the kind of activation pattern in the ventricular mechanics. I'm happy to be proved wrong, but the models take currently 24 hours to solve. So adding in full perfusion mechanics would be a big step for us.
And a related question, other than the cardiomyocyte, the interstitial matrix that the cardiomyocytes live in are a big determinant of what happens,
Yep.
and that's influenced by fibrosis. Have you modeled any of these issues where you have various levels of fibrosis or interstitial matrix?
In the starting, we do that in the atria, looking at patchy fibrosis or dense fibrosis in the atria, and that's mainly looking at electrophysiologic effects, including the effects of fibroblasts and electrophysiology, which we can do within the modeling framework we have. We can alter the end result of that remodeling on mechanics. So if you tell me that the fibrosis is causing the collagen matrix to be more isotropic or less directional, we can make the stiffness of the myocardium more isotropic. If you tell me that there's more collagen, I can increase the density of collagen. So we can alter the end result, but we're not kind of at the level where we're saying from this high-resolution image of change in structure of collagen, here's how that might alter the mechanics of the whole heart.
So I have two very practical issues acutely. One is the science that Dr. Dracos is involved in, and that's imaging the LVADed heart. Are you familiar with left ventricular assist devices?
Yeah, I've had some ideas about how you could do that.
Yeah, because we would be very interested in understanding the 3D mechanics of, now of course there's a lot of technical issues because of the artifacts that metal creates in a CT scanner, but
Yeah.
you know, we would be really interested in, you know, what you can model and see in terms of changes in the ventricular geometry and contraction with recovery.
It certainly sounds like you've got a fancy new CT scanner.
Yeah, we do.
And that may improve some of the hard beam artifact that you're seeing. So certainly we've seen less hard beam artifact on I think it's going to be the same brand, same Siemens scanner from the catheter wires compared to our previous Philips scanner, which is probably just a generation of scanner, not a brand issue. So that's been quite interesting. And also the software we have for tracking motion on the endocardium, that would map straight over onto a retrosophisticated CT. So that would work fine.
And my last question has to do, you know, one of the big challenges we have in clinical cardiology is predicting sudden death. And as you know, our current approach is to put a defibrillator into people as a way to really just cover all our bases. Of course, the vast majority of people won't ever use or require the device. You have very interesting electromechanical data here, and have you looked at how various morphologies and electrical activations might predict a susceptibility to ventricular fibrillation?
Yeah, so the area we're looking at that in was within CRT, and how pacing lead, the combination of pacing lead anatomy and scar might come together to lead to a combination of things that might be more proarrhythmic or not. So we have a 4-year follow-up on quite a few patients, so you need to have that follow-up time to get that incident rate so that you've actually got enough things to predict. And so that's what the data set that we're currently going through now. So we have MRI, I think, of 70 patients. We've got the 3-4 year follow-up now, and we're looking at kind of, can we link their anatomy and their scar and the activation patterns that we're predicting with their arrhythmogenic risk.
Great, thank you.
Other questions? All right, well, if not, we've definitely used our time. Let's thank Steve one more time. And a number of your meetings